Hi, this is Dr. Collins-Jones again, and this is segment 5, the final segment of my quick review of solution and dilution lab math video series. In this segment, we will discuss serial dilutions, parallel dilutions, and give a few lab math examples. Let's start with serial dilutions. Serial dilutions are dilutions done one after another, that is, in a series. The previous dilution is used to make the next dilution. The dilution ratio, that is, the volume of diluent and volume of analyte, remains the same in each successive dilution. The advantages of serial dilutions are they are really fast. You can quickly do a whole series of dilutions in a very short period of time. They use less original material because you are only removing a volume from the original sample one time. After that, the subsequent volumes come from already diluted samples. Serial dilutions are very easy to automate using liquid handlers. The disadvantage of doing serial dilutions potentially is that mistakes are propagated. If you make a pipetting error someplace in the middle of your serial dilution scheme, every dilution after that will be incorrect, especially if you're not recording all the volumes correctly. So in this first slide, I just wanted to introduce you to the flow or pattern of a serial dilution, and we'll build upon that in the subsequent slides in this particular segment. So we start with our undiluted analyte solution. All right. Remember, we're going to use the same volume of analyte solution each time and the same volume of diluent each time. All right. And so in this slide, I've already put diluent in the microcentrifuge tubes, which is commonly what we do serial dilutions in our labs. All right. So we start then by removing a specific volume of analyte solution, of undiluted analyte solution, adding that into our diluent volume, okay, and mixing it. And then once it's mixed, we're going to remove exactly the same volume, right, of the now diluted analyte solution from tube 2, pipette it into the diluent that's already present in tube 3, okay, and we're going to mix. And then again, we're going to remove the same volume of now diluted sample from tube 3, okay, and pipette that into the diluent already present in tube 4, and we're going to mix that. And then we're going to remove again an identical volume of the diluted analyte solution from tube 4, okay, and pipette that into tube 5 and mix. And you could continue this for as many times as you need to. Remember, the object here is to obtain a dilution that gives you a concentration of analyte in a range that is measurable by your measuring device, whether it's a spectrophotometer or whatever it is. Okay? All right. Let's start to then sort of look at how we would really set up a serial dilution in the lab. So the idea here is, oftentimes serial dilutions are already specified in the SOP, in the protocol, or in your lab manual. But suppose you're in the lab and you needed to design a serial dilution all by yourself for the first time. There are really two main things that you need to know before you begin making your serial dilutions. The first is you need to decide on a dilution ratio. For me, when I'm diluting an unknown solution, I have no idea what the concentration is. I've never made any measurements on it before. I usually go with a 1-to-3 dilution series because that gives me the widest range with the least amount of effort. Okay, and you'll see as we sort of go through the examples of different types of dilutions in the upcoming slides. So the first thing you need to decide on is what's the dilution ratio that you're going to use. The second thing that you need to decide on is what volume of the diluted sample is required for the measurement. So is it 50 microliters? Is it 100 microliters? Is it 300 microliters? Is it 1 mil? What's the volume of diluted sample that you have to have in order to put into your measuring device to make the measurement? And so obviously that means then you'll have to make more than that because you're going to take some out to make the next dilution. And that will become apparent as we illustrate this on this slide. So for the purposes of this slide, I've decided to choose a 1-to-3 dilution series. So every dilution will be a 1-to-3 dilution. All right, and then arbitrarily I've just chosen that you need 300 microliters of each dilution to make a measurement. So you have to have enough left over that there's still 300 microliters remaining in the tube that you're going to withdraw and put into the cuvette for the spectrophotometer or however else you're going to make your measurement. Okay? All right. So that means that to have enough solution to make the next dilution and to have enough remaining for the measurement. So there's two things that we need to consider here, right? We need to be sure we've prepared enough to make our measurement, but we've also have enough in that diluted tube to withdraw an aliquot to do the next dilution, right? So when we decide on things like this, we try to keep the numbers simple, right? We try not to get decimal points and things like that. I'll do an example on one of the slides where you do get a decimal point just to show you that it still works. It just looks weird. People don't normally do this, all right? So I've decided that we're going to make 600 microliters of diluent, of diluted sample in each tube, all right? And so the way this works now is I'm going to need to calculate how much of the analyte solution I need to dilute to 600 microliters, right? How much of the analyte solution I need to dilute to 600 microliters. So as always in this segment, we are going to treat things as a C1V1 equals C2V2 problem, right? And hopefully you remember from a previous segment that if we have a dilution ratio, we can treat that as a concentration if we're given volumes, okay? So C1 is going to be 3 and C2 will be 1, right? V1 is the concentration of analyte solution, and V2 is the final dilution, 600 microliters. And remember, for this problem, the 300 microliters is irrelevant because we were just told we need to have 300 microliters remaining in the tube in order to use for the measurement. All right, so again, C1 is 3 from our dilution ratio. C2 is 1 from the dilution ratio. V1 is the volume of analyte solution that we need to withdraw. And V2 is going to be 600 microliters, the final solution in the final volume in the tube. So solving for V1, V1 is 1 times 600 microliters divided by 3, or we need to remove 200 microliters of the analyte solution to do a dilution. And then how much diluent do we need to add? Well, that's going to be the final volume minus the volume of analyte solution, or 600 microliters minus 200 microliters, or we need 400 microliters of diluent solution. Okay, so just to summarize this, for a 1 to 3 dilution, we're going to mix 400 microliters of diluent and 200 microliters of analyte solution. Now, let's look at how we would actually set this up in the lab. So we have our undiluted analyte. This is what we're starting with. What I would do next, and I would call this tube number 1, okay? Just make sure that we keep track of everything. So then I would get 4 microcentrifuge tubes because I've just decided I'm going to try 4 dilutions. Hopefully that will be enough. I could do more, but just to fit everything on the slide and give you enough to show you the pattern, I've said we're going to do 4 serial dilutions. So we'll get 4 clean, dry microcentrifuge tubes, label them, right, sequentially 2, 3, 4, and 5. So now we have 1, 2, 3, 4, 5 in the tubes. And then what I'm going to do is I know that I need 400 microliters of diluent to add to each tube. So I'm going to pipette in 400 microliters of diluent into each of these tubes, 2, 3, 4, and 5. So it's already there to start with. And so that's easy because now you just set your micropipetter to 400 microliters and boom, 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 you add that into each tube. So you don't need to be changing volumes and everything else. You just add the same volume into each tube. All right. Now what I'm going to do is I need to add 200 microliters of analyte solution from the previous tube to the diluent in the next tube in the series. So obviously we're going to start with undiluted analyte. We're going to remove 200 microliters of that, add it into tube 2. We need to mix that, right? Because remember, the analyte solution is concentrated, the diluent is not concentrated, so there'll be 2 different densities, and so it will not give us instantaneous mixing. So we're going to mix this usually by vortexing for 5 or 10 seconds. So once we've mixed now and tube 2 is thoroughly mixed, we're now going to remove 200 microliters from tube 2 and pipette that into the 400 microliters of diluent in tube 3. We're going to mix it again and then remove 200 microliters from tube 3, add that to the 400 microliters of diluent in tube 4, mix, and then we're going to take 200 microliters out of tube 4 and add that into the 400 microliters of diluent in tube 5 and mix that. And that's how we do the serial dilution. And so, initially I had in here, right, 200 plus 400, right? So before we do the next transfer, there is 600 microliters in the tube. Then I take 200 microliters out, and that leaves 400 microliters in this tube, which is enough to satisfy our requirement for 300 microliters to make the measurement. Okay? All right. Let's go a little step further here and look at how we actually calculate dilution factors. Now, I'm still sticking with a 1:3 dilution. I'm staying with a 1:3 dilution, but I've changed the volumes around to show you that the volumes, the initial and final volumes don't matter as long as we have enough to make our measurement and we retain the ratio. So here I've said we're going to add 60 microliters of diluent to each tube, and then we're going to take 30 microliters of analyte from each tube and mix it with the 60 to give us 90 microliters, and that gives us then V2 is 90, V1 is 30, and that still gives us a 1:3 dilution ratio. All right, and so here's what this looks like. So how do we actually calculate then the dilution factor? All right, so there's no dilution for the neat tube. The next tube is 1:3, okay? The tube after that then, to calculate that, we're going to multiply the numerator, think of this as 3/1, by 3 each time. So 3 times 3/1, this is going to be a 1:9 dilution. Now it's going to be 9 times 3/1, or a 1:27 dilution, and hopefully you guys are now jumping ahead and saying, okay, this is going to be 27/1 times 3, or a 1:81 dilution, and then it'll be, right, 81 times 3/1, or 1:243 dilution. All right, and so that's how we get those dilution ratios. And whatever our initial dilution ratio is, that's what we multiply by. So let's just illustrate that on the next slide. So here, all right, I'm going to do 600 microliters plus 200 microliters. All right, so 600 microliters of diluent is added to each tube. The initial volume of analyte placed into each tube is 200 microliters. So the final volume is 800. So the dilution ratio is going to be 800 over 200, or 1:4. So our first dilution is 1:4, but now since the dilution factor is 4, this is going to be 4 times 4/1, or 1:16. Then this will be 16 times 4/1, or 1:64. And then 64 times 4/1, or 1:256. So that's how we get those dilution ratios. One more time just to illustrate it, because people always do the easy serial dilutions, which really doesn't give you the widest range of dilutions, but people often do a 1:2 dilution. And so we've added 25 microliters of diluent to each tube, and we're using 25 microliters of analyte solution. And so final volume before dilution, before we remove the aliquot for the next dilution, is going to be 50. So the dilution factor is 50 over 25, or a 1:2 dilution. So this is 1:2. The next one will be 2 times 2/1, or 1:4. Then it'll be 4 times 2/1, 1:8. Right? 8 times 2/1, or 1:16. Right? 16 times 2/1, or 1:32. And so that's how we calculate the dilution ratios. Okay, hopefully that was clear, and you guys can easily do that whatever you're given. Okay, next, what I said at the beginning here, I just want to emphasize. All right, we can have any multiple of the volumes that we use as long as the final volume, that is the analyte volume plus the diluent volume, divided by the analyte volume, is 1:4. So we could do 20 plus 60, we could do 80 plus 240, 100 plus 300, 5 plus 15. It doesn't matter because, if we have 20 plus 60, final volume is 80 over 20, that's 1 to 4. 80 plus 240, final volume is 320. 320 divided by 80 is 1 to 4. 100 plus 300, that's 400 microliter final volume divided by 100, that's 1 to 4. 5 plus 15 is 20, so 20 divided by 5 is 1 to 4. Right? So we've maintained the dilution ratio, but we can just make different multiples of the volume. And then I've just illustrated the same thing here. I'm not going to read through it. I'll just pause for a minute, and if you want to pause further, you can do that if you want to copy this down. But the key here is that we need to maintain the ratio of the final volume to the volume of analyte. Okay? All right. Let's move on then and go to the next level, and that is let's calculate the resulting concentrations. All right? So again, guess what? C1V1 equals C2V2, but in this case, the resulting concentration or the final concentration is C2. All right? So C2 is going to be C1 times V1 over V2. And so now, remember, we've diluted something. So the resulting concentration, if we dilute something, the resultant concentration is going to be less. It'll be smaller, right? So the concentration in tube 2 should be less or smaller than the concentration in tube 1. Okay? So V1 is always 30 and V2 is always 90 for our dilution series. And C1 is a concentration of the previous tube. So you sort of have to think about this. We calculate C2 for tube 2, but now that becomes C1 when we calculate tube 3. And we'll illustrate that as we go on. So we have a 4.00 milligram per mil starting concentration. That's our undiluted analyte concentration. Okay? And so that's tube 1 with C1. And now we're trying to figure out what is C2 for tube 2. So for tube 2, C2 is going to be 4.0 milligrams per mil. That's our starting concentration, times 30 microliters, that's our V1, divided by 90, which is our V2, or 1.33 milligrams per mil. And that's the concentration in tube 2. But now, this concentration in tube 2 becomes the starting concentration for tube 3, where we now need to calculate the concentration in tube 3 after we've done the dilution. So for tube 3, it's going to be C1 is 1.33 times 30 over 90, or 0.444 milligrams per mil. All right, and that's tube 3. And now the tube 3 concentration becomes C1 for tube 4, and we calculate tube 4, it's going to be 0.444 milligrams per mil times 30 over 90, or 0.148 milligrams per mil. And note that we're keeping our significant figures in the proper to the proper number of sig figs. So hopefully that makes sense, and this is sort of where we do the series here. The previous concentration is now C1 for the next concentration. Okay, so again, let's just do a little practice here, and if you want, maybe you would want to pause this and then try this calculation and see if you come back with the same result as I do. So I'm going to count to 3, and then I'll resume talking and talk through the video here. Okay, so we're starting now with 345 microliters of 7.25 milligram per mil starting analyte concentration. Okay, so the first thing to point out here is that that number, 345 microliters, it's irrelevant to the problem. It could be any volume as long as it's enough to be able to make the next dilution. So that initial volume is irrelevant. It doesn't matter in solving this problem. You can ignore it. Okay? All right. So we've done a series of 1 to 6 dilutions. We've said that our starting concentration is 7.25, and we're combining 250 microliters with 50 microliters, and so we have 300 microliters over 50, or our dilution factor is 1 to 6, and then you can check my numbers here to make sure that each successive dilution factor is correctly calculated. But our purpose here is to really calculate the resulting concentration in each tube. So tube 2, what's the concentration in tube 2 after we do the dilution? So tube 2 is going to be the concentration that was in tube 1, our undiluted analyte, times 50 over 300, or you could say 1 over 6, it doesn't matter, is equal to 1.21 milligrams per mil. Okay, and then now for tube 3, that 1.1 milligrams per mil becomes the starting concentration for making the dilution in tube 3. So it's going to be 1.21 times 50 over 300 equals 0.201 milligrams per mil. Hopefully you're already ahead of me on tube 4. Tube 4, right? The concentration that we calculated for tube 3 becomes the initial concentration for tube 4. And so we plug that in, and our concentration is now 0.0336 milligrams per mil. Tube 5, we're really starting to get some dilutions here. Tube 5 now is going to be 0.0336, which we had in tube 4. That's our starting concentration for tube 5. And again, it's just 50 over 300. And so we calculate 0.00559 milligrams per mil. And then in tube 6, right, the concentration we calculated for tube 5 now becomes the initial concentration for tube 6. And when we calculate this out, this becomes 9.32 times 10 to the minus 4th milligrams per mil. Usually when we get to those numbers that are more than 10 to the minus 3rd, we start to put them in scientific notation. That's common practice. But either way of indicating your answer should be okay. All right, so hopefully we now know how to calculate the resulting concentration in each tube when we do a serial dilution. Parallel dilutions, they're a little different. Here, individual volumes of the original sample are removed and diluted separately. The advantage of a parallel dilution series is that you're not propagating mistakes. Each dilution is unique. So if you make a mistake in one dilution, it does not affect any of the other dilutions. The other thing is we can make unique nonlinear dilutions. It's not a set ratio of 1 to 3, 1 to 6, whatever. It's not a set ratio, so we can sort of just make all different kinds of dilutions. The disadvantage is we're always removing material from our undiluted analyte, so we're going to consume or use up more starting material. And because we're not doing the dilutions in series, each dilution is unique. It takes longer to complete, and this is not easy to do using a liquid handler. So for an example here, I've simply said prepare a range of dilutions, each that has a final volume of 250 microliters. And again, I just chose that final volume arbitrarily. It could be any final volume, but that's what I chose for this example. Okay, so here we have our undiluted neat sample. We need to have 250 microliters as our final volume. So suppose I just remove 10 microliters of the undiluted sample. That means I need to add 240 microliters of diluent. Right? In the next dilution, maybe I'm going to take 20 microliters. So that now means I need to add 230 microliters of diluent. In the next one, I might decide that I'm going to dilute 50 microliters of the undiluted sample. So now I need to add 200 microliters, right? Final volume is 250. So it's going to be 250 minus 50 gives me 200 microliters. And then in the final one, I'm saying, okay, let's take 100 microliters of the undiluted sample. And again, I need to dilute it to 250. So it's going to be 250 minus 100 microliters, or I need to add 150 microliters of diluent. So you can see that this is a little bit irregular. Each dilution is independent of the other dilutions, but if you look at this, I've used up a lot of undiluted sample, right? 10 plus 20 plus 50, that's 80 plus 100. I've used up 180 microliters of my original sample, right? And so that's why oftentimes people sometimes avoid parallel dilutions. Okay. Another example here, prepare a range of dilutions, each with a final volume of 200 microliters. So I have my undiluted neat sample, all right, and now we're going to do 10 microliters. I need to prepare 200 microliter final volume, so I need to add 190 microliters of diluent. 200 minus 10 gives us 190. Next one I've said, let's do 20 microliters. So 200 minus 20, I need 180 microliters of diluent. All right. Then I'm saying, okay, let's do 50. 200 minus 50 means 150 microliters of diluent. And then I'm saying, let's do 100 again. And so now 200 minus 100, it's 100 microliters of diluent. Now, let's go a step further and calculate the dilution factor. So the dilution factor is V2 over V1. Okay. And so for our first dilution, V1 for tube number 1, V1 is 10, V2 is 10 plus 190, or 200 microliters. So 200 over 10, this is a 1 to 20 dilution. For tube 2, okay, this is going to be 200 over 20, or 1 to 10. For tube 3, right, our dilution factor is going to be 200 over 50, or 1 to 4. And for tube 4, our dilution factor is going to be 200 over 100, or a 1 to 2 dilution. Okay? All right. A little more complex here, and I think in this one I've given us some weird numbers just to show you that as long as you follow the rules for calculating dilution factors, they work, even if you get a weird looking number. All right, so here we're going to prepare a range of dilutions in which the final volume is greater than 150 microliters. So notice I changed this up a little bit. Greater than 150 microliters. It could be anything, right? But we don't want to make it too large a volume because we'll waste too much of our original undiluted analyte sample. So first one, 15 microliters. And I'm going to say, okay, I'm going to add 285 microliters of diluent. And the point of doing this sort of non-linearly is to just reinforce to you that if you make a mistake when you're combining volumes to do a dilution, as long as you record those volumes, you can always make the calculation. All right. So here I've added 285 microliters of diluent. So my final volume here is going to be 300 microliters. Here I'm adding 30 microliters of analyte, and I'm adding 170 microliters of diluent. And I've just picked the volumes of diluent at random. Okay. There's nothing special about them. Okay. And then 2.0 microliters of undiluted sample, 178 microliters of diluent, and then 250 microliters. Wow, that's a lot. Okay. And I'm going to add 750 microliters of diluent. So now let's calculate the dilution factor for each of these. Okay. So for tube 1, dilution factor V1 is 15, V2 is 15 plus 285 or 300 microliters. So it's 300 divided by 15, or a 1 to 20 dilution. For tube number 2, it's V1 is 30, V2 is 30 plus 170, or 200 microliters. And this does not turn out to be a nice, neat number. Our dilution factor is 1 to 6.67. This is not something you would actually try to do in the lab. You want to keep these nice whole numbers, no decimal fractions. But if you made a mistake and you kept track of your volumes, you could calculate the dilution factor and then back calculate to the initial concentration. Here we have V1 for tube 3. V1 is 2.0. V2 is 2.0 plus 178, or 180. So we have 180 over 2, or a 1 to 90 dilution. And then for our final tube 4, we have 250 microliters as V1, and V2 is 250 plus 750 is 1,000, or 1,000 divided by 250 is a 1 to 4 dilution, right? And so again, you can see parallel dilutions. You can kind of do any combination you want as long as you know the volume of analyte or original sample and the volume of diluent. But really, if you're in the lab, try to keep these nice, neat, whole numbers. Okay. Now, suppose we want to do a calculation and figure out what is the actual concentration in each of these tubes. Again, we said, okay, here's 16.2 mils of starting material, 16.72 mils of starting material. Doesn't matter how much you start with, right? You just have 16 mils of that material, which is good because you have a lot. You won't run out even after you've completed your dilutions. But what matters here is the concentration, the 15.7 milligrams per mil. So again, C1V1 equals C2V2, right? This is the same calculation that we did on the previous slides. It's just we're doing it with parallel dilutions instead of serial dilutions, but I just want to make sure that you know how to do the back calculation. So C2 is what we're solving for, and that's going to be C1 times V1 over V2. So C1 for every one of these tubes is 15.7 milligrams per mil, right? Because we're always removing our sample from the original tube. So every C1 is going to be 15.7 milligrams per mil. So now we just plug this into our C2, right? And our first tube is going to be 15.7 times 1 over 20, because remember, we are diluting it. So the ratio needs to make this number smaller. So there's tube 1. Okay. Next tube, C2 for our tube number 2 is still going to be 15.7, but now we are times 1 over 6.67, or 2.35. And notice again, we're paying attention to significant figures, 3 significant figures, because that's what was in our concentration over here, 15.7. Tube 3, okay, again, 15.7 times 1 over 90, okay, or 0.174 milligrams per mil. And then finally tube 4, C2 equals C1 times V1, 15.7 times 1 over 4, or 3.93 milligrams per mil. And I agree, this looks weird because we're not going in a nice sequential order of increasing or decreasing numbers, but this is just to show you the power. As long as you keep track of what you're doing, as long as you record your data properly, you will be able to calculate whatever you need to calculate. Finally, just 2 slides left. We're going to just give you some examples of lab dilutions. These are the types of things that we do in our labs. So the problem here would be prepare 850 microliters of a protein solution for SDSPH, which is 1.75 micrograms per microliter and 1.25x in gel loading buffer. The concentration of the supplied protein sample, that is the homogenate, which was determined using a protein assay such as the Kumasi Blue or Bradford assay or the BCA assay, was determined to be 18.3 micrograms per microliter, and the stock concentration of the gel loading buffer is 4.5x. So this hopefully is sort of a way to bring you back to some of the earlier segments and tie all this in. Just a final set of practice problems. So again, remember, we're going to treat each component separately, right? And both of these are C1 V1 equals C2 V2 calculations. So we'll do the protein. We have 850 microliters of the solution. The concentration of the protein is 1.75. That's what we want to end up with. And our starting concentration is 18.3, right? We have 2 concentrations. 18.3 is bigger than 1.75. So this gives us C1 is 18.3, C2 is 1.75. V1, that's how much of the 18.3 micrograms per microliter protein solution we need. That's what we're going to calculate. And then the amount of solution we want to end up with is 850 microliters. So C1 V1 equals C2 V2. 18.3 times V1 is equal to 1.75 times 850. Solve for V1 and we get 81.3 microliters of protein, and again 3 significant figures. To calculate the amount of gel loading buffer then, right, we have 1.25x gel loading buffer, 4.5x stock solution, and we're putting this in the same final volume of 850 microliters. So again, C1 is 4.5, C2 is 1.25, V2 is the same because these are being combined into the same solution. It's 850. Solve C1 V1 equals C2 V2, 4.5x times V1 equals 1.25x times 850. Solving for V1, we get 236 microliters of gel loading buffer. Again, 3 significant figures. But if you look at this, 81.3 plus 236 is not 850 microliters, so we are going to need to add some water. And so we always use ultra-pure water for our dilutions. So the amount of ultra-pure water to add is going to be 850 microliters, the final volume, minus the volume of protein, minus the volume of gel loading buffer, or we need to add 533 microliters of ultra-pure water. So into one single tube, we would add 81.3 microliters of 18.3 micrograms per microliter of protein, 236 microliters of 4.5x gel loading buffer, and 5.33 microliters of ultra pure water. All together, mix it up, and now we have 850 microliters of our protein solution at 1.75 micrograms per microliter and 1.25x gel loading buffer. Okay. So generally, we load 10 microliters of protein, that is 2.0 micrograms per microliter and 1x gel loading buffer per well. I just wanted to give you some sort of strange looking numbers to just show you that the process works. Use the pattern of calculation and you will come up with the correct answers. Last example. Determine the volumes of DNA, master mix, and PCR grade water required to prepare 125 microliters of a PCR solution which contains 0.025 micrograms per microliter of template DNA and 3.5 times master mix. So we're going to not just do 1x, we're going to do 3.5. The concentration of the isolated template DNA was determined to be 36.5 micrograms per microliter, and the stock concentration of the master mix is 15x. So again, treat each component separately. Still C1V1 equals C2V2 calculations. Calculate the DNA, right? And we have 0.250 micrograms per microliter of template DNA, but the isolated DNA, the starting concentration of DNA is 36.5. So plug that information into our C1V1, C2V2 variables, and we see that the first challenge we have is C1. We have 36.5 micrograms per microliter per mil. Look at that, 36.5 micrograms per mil, but our C2 is micrograms per microliter. So we need to make the volumes consistent, and I'm choosing to turn the C1 into micrograms per milliliter in order to make the volumes consistent. And we pipette a microliter, so I'm going to do microliters. Okay, so C1 in microliters is going to be 36.5 micrograms per 1 mil times 1 mil per 1,000 microliters, or 0.0365 micrograms per 1 microliter. So now we do our V1 calculation, and V1 is 0.025, okay, times 125 over 0.0365 micrograms per microliter, or 85.5 microliters of DNA. Okay, now let's calculate, and again, three significant figures, sorry. Now let's calculate our master mix, right? We need 125 microliters again. 3.5x is the final concentration of master mix, and the starting concentration is 15x. So plug that into our C1V1, C2V2 variables, right? Solve for V1, and V1 is 29 microliters of master mix, okay? And again, this time two significant figures, because really we're dealing with two significant figures here. Okay, and then finally, to calculate the PCR grade water, it's the final volume that we want, the total final volume. We want 125 microliters minus 86.5 microliters of DNA minus 29 microliters of master mix equals 9.5 microliters of PCR grade water. And that is the end of segment 5 and the end of this video series on lab dilutions, serial dilutions, and parallel dilutions. I hope this helped, and I will have some other videos coming up on significant figures and basic data analysis for the lab. Bye.